Hello, my name is Michael West. I'm the CEO of AgeX Therapeutics, and I'll be presenting here a corporate update at the Annual Meeting of Shareholders, December 2019. In doing so, if you look on the second slide of the slide deck accompanying this webcast, you'll see our forward-looking statements. So I'll be making statements that have associated risks and uncertainties, and so we refer all of our shareholders, investors, those considering investing in the company, to review our filings with the SEC for more detailed information about the company. Slide 3 describes the corporate mission of AgeX. We're targeting the largest and fastest growing markets in medicine. They really, truly are. Age-related degenerative disease. We all age if we live long enough. The unique demographics of the United States and many other countries around the world are leading to a really significant, the most demographically significant trend of our time, the aging of the baby boom population. And associated with that is not just mortality, but the late life of very high prevalence of chronic degenerative diseases. Chronic disease accounts for some 80% of the total healthcare expenditures of the United States, well over $2 trillion a year. The second bullet point mentions the fact that we're employing advanced technology, and I use this word advisedly, that reverses aging in cells and tissues in the body. And I'll be drilling down on that a bit later in the talk I'll give today. And induces regeneration in diverse tissues of the body, not just a particular tissue, but in many different tissues. So that's our mission. And of course, if we deliver on that mission, we're addressing some really significant market opportunities. I just want on the slide 4 here to give a little perspective on the revolutions in biotechnology, the history of those revolutions, because they directly relate to what's going on currently in this field we call regenerative medicine. So on the left on this slide, you see recombinant DNA technology. You know, you can almost date a person nowadays about whether they remember the advent of recombinant DNA. It was once considered very controversial. I remember it being on the cover of Time and Newsweek magazines. It was the ability to cut and splice DNA, and so we call it recombinant DNA technology. It led to some significant companies, indeed the birth of biotech itself. Genentech, Amgen are some of the early leaders in recombinant DNA. What could it do? It could allow companies to manufacture previously rare and valuable or unattainable proteins in a pure and industrially scalable form. So, for instance, insulin, instead of extracting it from the pancreas of pigs, could be made in a company on an industrial scale and very cost effectively in a pure form. Similarly, some years later, there was a revolution in the generation of monoclonal antibodies. These antibodies, of course, are proteins that circulate in the blood throughout the body and recognize a particular thing we call an antigen. It could be a virus, it could be, virtually anything the body would recognize as foreign. And monoclonal antibodies, the development of what are called hybridomas, transformed the industry and again allowed companies to manufacture these proteins called antibodies on an industrial scale and in a pure state. So we come to the third revolution. I was happy to say I organized the first effort back in the mid-1990s to isolate the first pluripotent stem cells. This was in collaboration with Jamie Thompson and John Gearhart and Roger Peterson. And these cells led to this birth of this new industry we call regenerative medicine. Like these previous revolutions, it takes about 20 years from the first discovery of the science and technology to significant commercialization. But now, having passed that 20-year mark, we're seeing, as this slide shows, major acquisitions now approaching billion-dollar valuations for company in this sector as we approach the time when products will be in the future approved by the FDA for many applications. Regenerative medicine delivering on, like the previous revolutions, the ability to manufacture, in this case, living cells on an industrial scale. Previously rare and valuable cells that could not be made available for transplant. So slide 5 shows the power of this platform of pluripotency. These cells are at the base of the tree of human cellular life and can become literally any of the cell types in the human body, making available a parts supply store, basically, for the human body. The ability to make young replacement cells of any kind useful in treating a wide array of degenerative diseases. And as I mentioned at the outset, the chronic degenerative diseases of aging are the largest single problem we have in our healthcare system and with the aging population, and the ability to manufacture young replacement cells to, for instance, regenerate heart muscle function and heart failure, or Parkinson's cells for Parkinson's disease, or cartilage for arthritis. All these kinds of applications can be transformative. This platform allows the uniform and infinitely scalable production of product, just like recombinant DNA and just like monoclonal antibodies. This is not true with technologies like adult stem cell technologies. These cells have a finite capacity to be scaled up and to make product. This platform also allows the precise genetic modification of the cells, which is now becoming increasingly recognized as critical to the future of regenerative medicine. For instance, you're seeing CAR T, a real revolution in cell therapy, usually directed toward cancer. Companies are now trending toward the use of pluripotent cells because this allows the precise genetic modification of cells to make smart cells, so to speak, of all kinds. And it also allows, because the cells can be precisely modified, off-the-shelf products, meaning cells that can be transplanted into anyone with reduced risks of transplant rejection, which currently accompanies cells and tissues that are transplanted between people. So let me walk you through the technologies which we're quite proud of that we've assembled over the years and brought into AgeX. First, a very large patent estate of over 400 patents and patent applications filed worldwide, mostly in this field of pluripotency. PureStem is a manufacturing technology which our scientists invented. Slide 7 shows the advantages of this platform. At the top, you'll see how virtually everybody else, companies and academia, making products from pluripotent cells, how they generate product. They take the cells and they induce them to become some cell type of interest. Let's say cells for Parkinson's disease, for instance, and then try to purify the desired cell types from what will obviously be a mixture of different kinds of cells. After all, there's over 1,000 different kinds of cells that these cells can make. And the problem with that, and it's a big problem, is that there are still impurities. And these cells that are contaminating the product are alive. You know, if you're selling a drug, like, I don't want to pick any particular drug, but let's pick Tylenol for no particular reason. Tylenol might have some impurities, it may not, but if it did, you know, your body would excrete the impurities or metabolize them away or deal with it. If you transplant cells into a patient that have impurities, where the impurities are living cells, they can grow and make an undesired type of tissue of where you're transplanting cells. And so that would be undesirable. And that problem is a major challenge for companies historically in making products that can be approved by the FDA. This is addressed beautifully with our PureStem technology. PureStem is shown at the bottom of slide 7. Here we take one cell, a single cell, that is on its way, it's no longer a pluripotent cell, and it's on its way to becoming this or that type of cell, like a heart muscle cell or a cell for Parkinson's or whatever. And that cell is expanded, as we say, clonally, from a single cell into a population of millions and millions of cells that can be scaled up in an industrial scale, but they're 100% identical. We call it PureStem. It's our trademark for the product or the technology. And we have shown in the past that we can make over 200 different cell types of the human body using this, and we easily skirt around these manufacturing challenges that the rest of the industry faces using PureStem. This is a very strong advantage that we have as a company. Okay, slide 8 is another part of our technology platform that we have called UniverCyte. This is using HLA-G as a cloaking device, in a sense, to allow cells to be transplanted with reduced risks of transplant rejection. Slide 9 just summarizes some of the ways HLA-G works. First, its primary role in nature, in humans, appears to be to suppress the mother's immune response to the developing baby. So, as many of you may know, the baby is not genetically identical to a woman. Indeed, a woman can carry a child from completely different parents, a surrogate mother. She does not reject, her body does not reject the cells from the baby as foreign. And we believe that the critically important in this cloaking or protection of the pregnancy from rejection by the mother is this molecule called HLA-G. And we have patents that now from in the U.S. and other countries protecting the use of our patented technology to engineer our pluripotent stem cells to then make products, all of which express this gene on the surface. And the evidence is that HLA-G can disarm multiple different arms of the immune system to help prevent rejection of transplanted cells. So in slide 10, this sort of summarizes our business model in cell therapy. This now allows, just like the previous revolutions in biotech, recombinant DNA and monoclonals, that's what we call it here, traditional biologicals. It allows production in a centralized facility of our, in this case, of our UniverCyte derived cell therapy products. Cells can be frozen and remain viable, and so they can be distributed as frozen inventory, thawed, and injected at point of care, just like many other biologicals. Slide 11 points out an additional technology we bring to the table in our regenerative therapies, and that's HiStem. I won't go into great detail about HiStem. There's additional information available online, but think of it as if the cells are sort of bricks in a wall, and we're patching up a damaged wall, for instance. We're injecting heart cells into the heart, for instance. The bricks would need a mortar to hold them in place. Cells in the body have an extracellular matrix, it's called. It's like the mortar that holds bricks in place. And HiStem is a synthetic matrix patterned after natural molecules of the extracellular matrix, but it's designed to cross-link like mortar would harden after bricks are put in a wall. HiStem hardens in the sense that it cross-links with cells, enabling the stable engraftment of cells at the intended site of delivery. All right, slide 12, our business strategy. How are we going to make business with all these amazing technologies? Well, I'm going to first discuss the bailment of our master cell banks. Slide 13 sort of shows us how this will work. We have at the company CGMP compatible, good manufacturing practices compatible human pluripotent stem cells. So these are cells that are capable of making all of these different cell types, and they were manufactured in conditions to allow them to be used, the products from them to be used in human clinical use. So they were derived under these GMP compatible conditions, unlike many of the other cell lines out there. Now, what we plan to do is engineer these cells, for instance, with this HLA gene modification. So we now have these cells capable of making off-the-shelf products, and then bail them or rent them out to various other biotech and pharma companies. We will own the master cell banks under these agreements. We'll continue to own them. When the licensee makes a particular cell type, let's say they made, for instance, heart muscle, they would then own and could sell the heart muscle. But the original master cell banks we continue to own, and that rental agreement is a royalty-bearing agreement. Now, it's known in the industry that once you have a registered product, especially with cell therapies, where we don't think it's going to be easy to make a generic or a biosimilar product, that companies would be very reluctant to change the cell type because that require repeating all of the potentially preclinical as well as clinical studies that led to the registration of the product. So it's very likely, we believe, that our customers will continue to use this cell bank indefinitely into the future. If you look at the net present value of royalty streams, especially where you can have multiple, hundreds potentially, of different products made from these cells, and having the royalty streams run into the indefinite future, unlike a patent license where typically royalties cease with the expiration of patents, this has significant net present value. In addition, slide 14 shows other uses of the cells. We plan to maximize the commercialization of this platform. Of course, our PureStem lines and our other products such as biomaterials made from these cells, or organoids, which are simple precursors to various organs, or products to be made for drug discovery. All of these are potential commercial avenues for the company in developing products from these cells. Slide 15 shows our plan to partner some non-core applications. and those include the therapeutic use of ectopic organoids, diverse biomaterials, including exosomes. For those of you who know, exosomes are an emerging field of therapeutics. They're small, spherical entities that are sort of a natural counterpart to viruses, and they carry messages throughout the body, and people are interested in developing those as novel therapeutics. Other biomaterials that our cells make are all the various kinds of proteins that are secreted by cells, and there are hundreds of those. Lastly, Cytiva II. These are cardiac cells used in drug testing, but you notice I say adult cardiac cells. Let me explain what I mean by that. Slide 16 shows, this is a slide from GE Healthcare. We formed a partnership, a collaborative agreement, license agreement with GE Healthcare in the past. By the way, GE Healthcare is anticipated to be sold to Danaher Corporation, and our contract will then be with Danaher. In this agreement, we have license to the relevant patents of GE Healthcare in this field, and rights to the trademark Cytiva, as well as the significant amount of data they've accumulated relating to Cytiva I, or Cytiva. GE Healthcare initially entered this field recognizing that many products, such as the ones I'm showing you in slide 16, failed because of cardiotoxicity. Now, how could that be after drugs go through all this testing? So, you know, drugs are tested diligently before they enter clinical trials, and then, of course, in clinical trials. But many of the preclinical studies are done in animals, for instance, in rodents like mice. And mice are not humans. There's fundamental differences. And many of these drugs, and these can account for billions of dollars of losses to industry when a drug goes through all this years worth of research and development and has been perhaps in, for instance, in a phase 3 clinical trial, or is already approved and then is pulled from the market because of cardiotoxicity. How could this not be seen in an animal study? Well, mice are not humans. And so GE recognized that being able to make heart muscle in the dish solves numerous problems for the industry and potentially helping to solve this arguably billion-dollar problem that they have of unanticipated cardiotoxicity. So how do you make heart muscle cells in the dish? Well, attention got drawn to pluripotent stem cells. Now, the problem with pluripotent stem cells is that heart muscle made from them are still in the early stages of development. They're not yet adult-like. So heart muscle doesn't become adult-like actually until after you're born. And nevertheless, industry is using these immature heart muscle cells. Well, based on our, I think, significant understanding of the aging process, we have been able to identify ways to mature these cells in a very important manner, and that product is called Cytiva II. We filed a patent application on this technology, and through our relationship with GE Healthcare soon, Danaher will be completing the development of this product and launching product as early as the coming year in 2020. In addition, of course, slide 17 reminds us we'll be working on our internal development of AgeX BAT1 and VASC1, and I'll quickly review for you the nature of those products. BAT1, brown adipose cells, have attracted really significant interest in the public and in medicine. If you go to a checkout line at the counter, this is how I came to recognize how important these cells were. One day I was at a checkout line at the grocery store and saw every single magazine around me dealt with weight loss. And especially here as we look at the turn of the year and the new year, everyone's thinking about making their resolutions on weight loss. Well, weight loss is tough. As slide 18 shows, one solution on the right is exercise. On the left, you can cut caloric intake. The problem is people find it hard, and it doesn't work. There are significant reports by epidemiologists and people in the field of nutrition that are just quite candidly say, it can work, but it doesn't work because people don't do these things. They don't exercise properly and they don't cut the caloric intake. And so we need a new solution to this problem. Now, what could that be? On slide 19, we show a recent development in medicine. I mean, recent within the last 10 years or so. It's come to be recognized that there's a tissue in the body called brown adipose tissue. It's called brown because it's a different kind of fat tissue. Fat, of course, stores calories for future use. It's the bad kind of fat. Brown fat is the good kind of fat. It actually burns calories rather than stores them. It disposes of them. Its function is normally multiple in nature. Brown adipose tissue is the major source of heat production other than shivering when you're cold. So it's sort of a furnace, and it pulls sugar and circulating fat out of the blood, and it literally generates heat to warm the body. But it does many other things, which we discuss in one of our scientific papers, which you can find available online. Now, on this graph in slide 19, on the left you'll see, this is from a published paper, not our own, showing the age-related loss of this tissue. And you see how precipitous the activity of this tissue is lost with age. And then on the right, it's showing you the coincident accumulation of body fat content, which I don't need to remind any of the listeners in this webcast to. While we were young and we had a lot of this brown fat to balance our metabolism, we could eat all of those French fries and hamburgers. As we get old, even avoiding a lot of that food, we start gaining central obesity, and most seriously, the sequelae associated with type 2 diabetes could be things like coronary artery disease, where fat is stored in the coronary arteries and as an abnormal site of deposition of this excess fat. And so in recent years, it's come to be recognized that if we could restore brown adipose tissue to a youthful level, we may help reestablish that healthy balance we had when we were young. Animal models suggest that this kind of therapy could be efficacious. And so the question is, can we find a way to manufacture these cells on an industrial scale? And here we see in slide 20, tissue-sourced brown adipose cells. So here, brown adipose tissue was extracted. The cells were put in the laboratory dish. And as you can see, stained in red are the brown adipose cells using a marker there called UCP1. A minority of the cells are the active type of cell. Like all transplant products, sourcing cells and tissues from humans is challenging for many reasons. Ethical reasons, quality control reasons, cost of goods being a major issue. On the right, you can see that our PureStem technology can be a source of these cells, and because these cells are made from pluripotent cells, they can be made off the shelf, UniverCyte, universally engraftable cells. They can be made through PureStem to be, as you can see in the picture, pure, and they can be made industrially scalable with a very attractive cost of goods. And so we think this is a very exciting application of all the technologies I've described today, especially for one of the larger problems we have in aging, which is type 2 diabetes and all its negative consequences. Slide 21 reminds us of the severity of the problem. 30 million Americans have diabetes currently. 1 to 3 Americans will have diabetes. It's anticipated by the year 2050. Perhaps upward of half having at least pre-diabetes, which can have negative health consequences. And the markets, of course, for diabetes and obesity are extremely large and growing. And these cells are, unlike drugs, are a natural solution to this problem. These are normal cells that we lose with age, added back to restore healthy metabolism. That's the concept of the product that is currently under development at our company. Slide 22 shows us the nature of VASC1. These are, as I said at the outset, two of literally hundreds of cell types we could make from pluripotent cells. But based on our analysis of competition and market opportunity and many other factors, we've chosen these products as lead products for internal development. VASC1 addresses the #1 killer in the United States and many other countries around the world, vascular disease. So these are young vascular cells. On the slide 22, top left, you see PureStem vascular endothelium. These are the cells that make the vascular tube. On the bottom, below the slide saying monoclonal endothelium, you see GFP endothelium. These are our PureStem cells making vascular tubes in the dish, showing you their capability to form blood vessels. Now, the other images here in this slide are rather technical in detail. If any of you are cell biologists, you'll recognize the highly purified nature of these cells. They're showing absolute signature that these cells are what they appear to be, and they are in a very highly purified state. Now, what can we do with VASC1? Slide 23 reminds us that cardiovascular disease is approaching a $1 trillion market in the United States alone. Now, the problem with aging is impaired blood flow, primarily to the heart, but we see it also in the brain with ischemic stroke, peripheral artery disease, vascular insufficiency, and many other tissues in the body. But in the case of coronary artery disease and its associated heart attack or myocardial infarction, you can have coronary artery bypass grafts, you can have stents put in. We imagine that VASC1 will have profound applications in numerous age-related diseases associated with vascular insufficiency, heart, myocardial infarction, and heart failure being foremost on our mind. Now, slide 24 introduces us to a technology which is the most provocative technology which we've developed. we believe we're the intellectual property leader in. My personal prediction is that will be the most talked about new medical technology of the third decade of the 21st century. We call it induced tissue regeneration. It's called partial reprogramming by other scientists around the world. What is this technology? Slide 25 is an example of some of the published papers by other researchers. On the bottom left is a paper that was published in the journal Cell by a group at the Salk Institute. Its title is In Vivo, in other words, in the body, amelioration of age-associated hallmarks by partial reprogramming. That is essentially what ITR is. On the bottom right, you'll see a paper published by the group at Harvard Medical School with the title Reversal of Aging, which is a profound statement for a gerontologist to make. In this case, the senior author of this paper is David Sinclair. Reversal of aging and injury-induced vision loss by TET-dependent epigenetic reprogramming. And again, he's speaking of, in this paper, partial reprogramming, or what we call induced tissue regeneration. But what is this? Slide 26 summarizes the nature of the technology. About a decade ago, at our previous company that our scientists are working at called Biotime, we showed that it was possible to take cells from a patient and by using certain reprogramming factors, we could take those cells back in time, reversing the developmental age of the cells back to the very beginning of life. And so on the left of slide 26, you see rejuvenated primordial cells. These are cells like skin cells, for instance, taken from a patient, taken back in time, such that you could make young, again, heart muscle or cartilage or skin or bone or blood or whatever, identical to the patient, the cells they had when they were born. And we showed this in this paper that this technology reversed the aging of the cells developmentally, but also the telomere clock of cell aging, the clock that governs the lifetime of cells. Now that was with cells in the laboratory dish. That's commonly called induced pluripotency. The cells are called induced pluripotent stem cells. And we recently announced that we had filed, a previous company, but some of our scientists had filed a patent which recently issued on induced pluripotency, the patent having been filed before the now famous paper by Shinya Yamanaka, for which he won the Nobel Prize. So this is really a significant technology, but I believe that the most significant application, and again, the application which will be the most talked about new medical innovation in the coming decade, in my opinion, will be the application of this same technology in vivo, in the body, not just on your cells in a laboratory dish. Well, how would that work? Well, in slide 26 on the right is a very simple cartoon to show how this would work. In this case, the same factors are introduced into the body, and we have filed patents on numerous ways of introducing these factors into the body, including gene therapy and the use of mRNAs and other techniques as well, including the use of exosomes to carry these messages throughout the body. What the accumulating data suggests that this can accomplish a reversal of age of cells in your body, taking the clock backwards in time, but also inducing a regenerative state in the body, something that we tend to lose with age. In the first few days, for instance, after a human is born, if you had a heart attack, your heart would have the capacity to regenerate damage from the heart attack. After a week or two after you're born, if you have a heart attack, and of course most of us would have a heart attack when later in life, your heart cannot regenerate. It merely scars as a failed attempt to regenerate. And you see this throughout the body. The brain cannot regenerate. You get scar tissue in the brain and so on. And what this technology also does is unlocks that regenerative state. And that, of course, directly impinges on my opening slides about the immense need for technologies to allow the body to regenerate in the context of chronic age-related degenerative disease. So ITR, the ability to literally reverse the aging and induce a regenerative state in aged patients, we think is groundbreaking. We believe we have a very significant intellectual property estate, having filed early patents in the field. And to develop this aggressively, we've set up a subsidiary called Reverse Bioengineering. Slide 27 discusses this subsidiary. It's currently 100% owned by AgeX. We anticipate that we may fund Reverse Bio through equity financing in that company. There are profound cancer applications to ITR, which I have not discussed. I would say that the cancer applications of the technology are very significant, similar in potential impact to the use of the technology in some applications in regeneration. So very significant cancer applications, which we would plan to license out. We're not a cancer company. And then my last bullet point, since the technology applies to virtually every tissue type, we may partner out specific clinical applications of this. So for instance, in the eye or for the inner ear, for bone regeneration or heart regeneration and so on. And then retain certain key applications for ourselves. So slide 28 discusses some of our 6-month milestones for AgeX. We plan to do partnerships, including the use of our organoid technology, the commercial bailment of our master cell banks, which I described early on, PureStem-derived biomaterials. We plan to complete the development of our Cytiva II product. This could be, of course, this would be a research product sold to large pharma companies. So this could be a significant source of revenue in the very near term. And we have staff on hand prepared to market that product as aggressively as we can. Also, in our milestones for the first half of the coming year is the financing of Reverse Bio to aggressively advance this very important technology we call ITR. Slide 29 reminds you of some of the quick facts about our company, which can be found online. With that, at slide 30, I'll just summarize by saying we're targeting some of the largest unmet needs in the United States, the chronic degenerative diseases of aging. We plan to partner the non-core commercial applications of pluripotency and ITR, early commercialization through the sales of Cytiva II, and the bailment of our master cell banks can benefit the company by providing business opportunities in the near term. We plan to aggressively pursue the internal development of AgeX, BAT1, and VASC1 from our genetically modified master cell banks. And then lastly, keep your eye on induced tissue regeneration technology for transcription reprogramming of aging in vivo, which will be developed in our subsidiary Reverse Bioengineering. With that, I would like to thank all of you for joining us here today, and for those of you joining us online, we look forward to giving you future updates as they come along. Thank you very much.